Welcome, everyone. We'll give it just a couple more minutes for attendees to filter in, and then we'll get started. All right, let's go ahead and kick it off. Thank you for joining us. My name is Nicole Nolan. I'm the programs manager of Innovone Collaborative, and welcome to our seminar series. Thank you for attending. I'm going to cover just a couple brief housekeeping notes before I hand it over to Seppa. First and foremost, we want to thank our partners, the Chan Zuckerberg Initiative, Han Jean and Kim Jeans, whose generous support enables our mission, including this program, the seminar series. So I will be dropping my email in the chat, and if you or your organization are interested in discussing how you might support our mission, I would love to have a conversation with you. Secondly, I wanted to let you all know that we've built a new platform called Connections designed to support collaboration across the individualized medicine ecosystem virtually. And we will be continuing discussion from today's seminar on connections. So if anybody has follow-up questions or thoughts afterwards, we invite you to join us there. That's another link that I will drop in the chat. And that's all from me. So Seppa, may I hand it over to you? You're on mute.
Hi everyone, my name is Seppa Hermans. I will also quickly go over some details. We want this to be an interactive session, so if you're able to turn on your camera, we would greatly appreciate this. If you have any questions during the presentation, please put them in the chat and we will go over them after the presentation. This session is recorded. You can rewatch the session afterwards on Connections. After the session, you will automatically receive a survey. If you are able to, please fill in the survey. Here you can also give us feedback on how we can further improve our seminars and what other topics would be interesting to cover. And I will now introduce you to our speaker of today. Dr. Zohar Pode is a postdoctoral fellow at Harvard's Wyss Institute. She's currently working on innovative delivery platforms for oligonucleotide therapeutics. But prior to joining Harvard, she was employed as head of CMC and manufacturing at Aummune Ltd. This is an immunotherapy company, and here Zohar led the development of individualized aptamer-based platforms for solid tumors, advancing them from R&D to phase 1 clinical trials. And in today's talk, Zohar will highlight the development of an individualized immunotherapy platform using oligonucleotides for solid tumors. Zohar, you can take the floor.
Thank you. I'll share my screen. Okay, do you see my screen?
Yes.
Yes, okay. So good afternoon or good evening, everyone, depending where you are. My name is Zohar. I have a PhD in chemistry, and I want to talk to you today about CMC aspect of individualized platforms. That was part of my work at Aummune for 5 years. So a few disclaimers. Just 1 second. A few disclaimers to start with. So I'm currently a postdoctorate research fellow at the Wyss Institute, but the work I'm going to present today is not related to anything I do at Harvard or at the Wyss Institute. It's only related to the work that I did when I was at Aummune, which was from 2019 to 2024. I was their head of chemistry, head of CMC, and then also their GMP site manager. And another thing is that Aummune had 3 pre-IND meetings with the FDA regarding our platform, and we got a lot of feedback. We had a lot of correspondence with them, but eventually this first-in-human phase 1 clinical trial was approved in Israel. So eventually we submitted an IMPD, and the final submission was done in Israel. So although the FDA had commented on everything that I showed you, I'm going to show you today, the final approval wasn't done in the USA, but rather in Israel. And of course, all other regular disclaimers. So just starting with Aummune and what Aummune is aiming for. So Aummune is proposing a new approach for developing potent individually tailored drugs. So when talking about traditional drug development, first we had it plus something like traditional chemotherapy, in which one size fits all. We have one drug for all kinds of patients. The next generation was something we called precision therapy, in which physicians tailored individual treatments from existing drugs. So taking all kinds of drugs, if we talk about oncology, so all kinds of anti-cancer drugs, but then generating unique combination or specific, or using specific drugs for specific profile of patients or profile of tumors, but the drugs are already made, they're already approved, and they're known. But what Aummune is trying to generate here is first of its kind, first class therapy that is actually manufactured for each individual patient. So these are drugs that are not existent until now. We don't just take off-the-shelf drugs and trying to tailor them to specific patients. We actually manufacture new unknown drugs for each patient based on its specific tumor. So Aummune is working on the field of synthetic oligonucleotides. Basically, we are working with aptamers. I don't think I need to explain what aptamers are for this forum, but I will note 2 things. First is that aptamers are oligonucleotide sequence that their function is derived from the 3D structure, not from their genetic cargo. And the second is that the aptamers that we are working with are functional oligonucleotides, meaning that they don't just bind the cells, which is what usual aptamers usually do. We actually look for aptamers that has all kind of activity, and in this case, anti-tumoral activity. So they don't just bind the tumor cells, they also induce some kind of death to them. So how is it done? Aummune takes 2, wait, maybe I'll just add my pointer here. Pointer. Oh. Okay. So at Aummune, we take 2 samples from each patient. One of them is a healthy, usually blood samples, and the other one is a biopsy from the target, which are cancer cells. And first we take these target cancer cells and we introduce them into a random library of aptamers, usually around 10 to the 15th. And we are taking them through a select process, meaning that first we take all of them and we look for the aptamers for the sequences that best bind these cancer cells. And then we take them, we enrich them with PCR, and we take all the sequences that came out of this PCR. And now we have a smaller population, around 10 to the 8. Now we take all these sequences that are actually a group, they are a pool of aptamers. And we induce them again, and we introduce them again to the target cancer cells. But this time, we don't look for binding. We rather look for functional enrichment. So we look for the ones that best kill these cancer cells. And then again, we take the one that best induce killing to these cancer cells, and we enrich them, and we take them, and now we introduce them to the healthy samples. But now we are doing negative select. So we are looking for the ones that do not induce killing onto the healthy cells. And now we do a number of iteration between the negative and the positive selects, every time enriching the population that we are getting. So we are ending up with a pool of sequences, around 100, 200 sequences that have the best killing effect toward the specific cancer cells of this specific patient, but do not affect this specific patient's healthy PBMCs. We then take this, we sequence these sequences, and we individually synthesizing them. And we start by testing them one by one, and then we start to see which of them has the best activity and the best safety profile. And we ended up with just one aptamer that we are choosing that has the best killing effect towards the cancer cell of this specific patient and the least toxicity to its healthy cells, and also provided us with the best safety profile in our safety battery tests. So this is our final asset. We can see here the variable domain. This is the part that I just explained. This is the aptamer that induces killing effect on the cancer cell. This is our personalized part of our test item. We then take this part and we link it to another aptamer that binds and activates T cells. And this linking is done by CpG hybridization domain. So basically, we have a T-cell engager, which is not only a binder, it's a bispecific T-cell engager that binds and activates T-cell on the one hand, that binds and kills cancer cells on the other end. It's safe towards PBMCs and other healthy tissues, and it has three mechanisms of actions: T-cell engagement and activation, APCs stimulation, and direct cancer cell killing effect. We were able also to show these three modes of actions: the T-cell engagement, the APC stimulation, and the direct killing effect for each one of the aptamers that our platform produced. So our cycle goes like this. We take these two samples from every patient that enters our clinical trial, and we propagate its cancer cells by growing organoids until we have enough cells to work with, and they're all cancer cells. We then introduce these healthy cells and the cancer cells into our efficacy-driven selection platform that ended us up with one aptamer that will kill this patient's cancer cell, but will still be safe. We bind it to the immuno part of our system. We manufacture this specific and unique drug product for this specific one-of-a-kind patient, and we administered it back in a hospital to the same patient. For preclinical data, this is just one example that we were able to show that if we take tumor-bearing mice, this is a CT26 tumor, and we treat this mice as if it's our own patient. We take this cell line and we introduce it into our platform, and we find one aptamer that best kills these cells. And we do the negative selects for this mice splenocyte. So we ended up with an aptamer that kills this tumor but does not affect the splenocyte of these mice. Then we are able to get, when injected into established tumor, a huge reduction in size, even disappearance of almost all the tumors in the treated group comparing to the untreated group, and a very big difference in the survival. And if we also do another preclinical data that was done with 41, we were also able to show that this is a cold tumor, that we can actually turn it into a hot tumor by transforming the treated mice from the same aptamer that now was the part of a platform that goes through the 41 and not from the CD26 platform cycles. We were able to actually change the landscape of the immune cells in the tumor microenvironment with much more activators and less suppressor cells in this mice anti-tumor tumor microenvironment. So this is our individualized therapy, and this is why I introduced the precision therapy. So if you take precision therapy, in which you take a known drug and you just introduce it to a specific patient, or even a combination of drugs, these drugs are already approved and they are known and their safety profile was established. But when we are trying to actually develop new drug for each new patient, this is actually a much more challenging process, because we actually need to do this whole development process that you do for any new drug for any new patient that came across our platform. So some of the challenges that I want to introduce today, I won't show all of them today, but I will just go over some of them to give you a better concept of what kind of challenges individualized therapy has to face. So first of all is how to provide a proof of concept for efficacy. How many in vivo models do we need to do? Which species? How do we prove that individual therapy that needs to work in human actually going to work based on mice model? How do we de-risk the chance of off-targets effect, and how do we provide safety data? Because whatever sequence we are going to test right now, this will not be the sequences that we're going to be used in clinical trials. And in the same manner, every new sequence that is going to be used in the clinical trial that humans are going to be introduced with are not the sequences that went through the GLP tox. So how do we provide safety data for new sequences each time? What kind of data should we show the regulator? How to incorporate final oligo design to the selection process. Usually when you're trying to, I know you know better than me, when you're trying to develop an ASO or an siRNA or an aptamer or any kind of oligo therapy, you usually start with the naked one and you look for the best sequence, and only then you start to introduce protecting groups and see how do they affect its activity, its safety, its stability, its off-target effects, the protein binding properties of it. But now we're going to have new sequence every time. We can't just start doing all these tests over and over and over again, because then we won't fit into a reasonable time frame for the development. How do we design a pivotal GLP tox study? Again, this is the same problem. No matter which sequences we're going to use for the GLP tox, this will not be the sequences used in the clinical trials and later on. So what kind of GLP tox design would be acceptable for the regulator to do a proof of concept to show that our platform is safe, even though the sequences are going to be different every time? Which bioanalytical method can we develop if we're going to have a new sequence every time? Which kind of reference standards can we use, because we're going to have a new sequence every time? What kind of manufacturing and control mechanisms can we introduce, can we establish if we're going to have new and unknown sequences? So every time I'm going to synthesize a new sequence that I don't know exactly how it's going to behave in synthesis, in purification, in QC. So how do I even release this kind of oligos? How do I make sure that I'm going to meet the right set of specification? What is the right set of specification? What is the purity that I want to reach for new unknown sequences? And what does it even mean to have a purity for new sequences when they have unknown sequence, they have unknown impurities? So what does it mean to have any kind of qualification for their impurity or impurity levels, which was done for other sequences? And eventually, which is the most important part of this whole process, is how to design a process that will meet all these required criteria within a reasonable time frame for a phase 1 first-in-human clinical trial for immune oncology. This means we are going to get a very metastasized oncology patients, and the time frame that we had set for ourselves for our first in-human phase 1 clinical trial was up to 12 weeks. So around 3 months, we have to do this whole iteration from getting a biopsy, growing organoids, introducing it into the platform, identifying a new aptamer, manufacture and release the entire drug product, and then go back to the patient and administer this drug to him or her. This is when usually even just the manufacture and release of a single drug takes much, in a GMP environment, takes much more than 12 weeks. And we want to do this whole cycle in this time frame. So I'll start with the sequence selection process. I won't go all the challenges that I just talked about because it's going to be a very long talk. So I'll just say a few words about some of the more CMC related issues that we had. So for the sequence selection process, this is what I showed before. We have this selects process in which we start with a library of aptamers and we enrich and enrich and enrich, and then we synthesizing them in the individual manner. So we started by how do we improve the safety of this process. So we thought maybe we will need to qualify the specific library. We will take a library and we will use only this library and we'll know that it's safe and we will characterize it. But then we encountered all kinds of cross-contamination across different patients. And we had some sequences that are undergoing very good PCR rather than other ones that are starting to take over the whole population. And we were very concerned about this and about cross-contamination with just small amount of leftovers because PCR is very sensitive. So we actually ended up with the opposite conclusion for this process. So for each select process for a new patient, we actually start with a new library that contains unique primers that are not cross-reactive with any other primers used for previous patients. So we have on the shelf a number of libraries that we pre-made because the libraries are not patient dependent, they can be ready, that prior to them, we designed their primers to be unique and not cross-reactive between different libraries. Also, for the computational part of our selects, we actually limited the amount, the size of the aptamers to be constant. So we always get the same size of aptamers, which will make the development and then synthesis and all other manufacturing much more easy. And we restricted also certain motifs like GG repeats or other things that we know are going to be much harder in terms of manufacturing and also can lead to a lot of false positives later on. When we moved to the individual aptamer synthesis, the first thing we did was we bought a multi-synthesizer. So we took actually this activity in-house because, again, one of the most important factors in this whole process was the time. So we ended up with a possibility to actually do this whole manufacturing of candidates within a weekend. We could get the sequences from the sequencing team on Thursday and then introduce them into the synthesizer, process them, put them in the QC, and have everything ready and lyophilized by Sunday, which is like Monday for Israel. One more thing, and this is what I also discussed about, that usually when people do oligonucleotides development, they start with a naked sequence and just check the sequence, and then all other modification and protecting groups are screened after the final sequence is already established. But this time we had a problem because we didn't want to choose the sequence and then start adding the protecting groups and then see that we're going to have a problem with this. And this is especially true for aptamers, that their activity is derived from the 3D structures. So anything that will change this 3D structure or will change the way it interacts with its target protein may affect the activity. So what we did was we screened several candidates for several protecting groups, and we chose the final protecting groups and modifications we are going to work with. And we actually introduced this to the selection process. So by the time we're synthesizing the individual candidates for selection, they are already being synthesized with the final protecting groups. And like with the final modification that the final drug product is going to have, so we can test them and screen them and choose between them the way they're going to look in the final drug product. GLP tox design and bioanalytical method. So this was, again, a huge challenge, and we had a lot of discussion with the FDA regarding what kind of GLP design, GLP tox design will satisfy them in terms of to show that the platform is safe enough. So eventually we ended up with this design in which we have a pool of 5 test items. So this is actually 5 different drug products. All have the same T-cell engagers, so that binds a T-cell because it is identical across all patients. And we also proved that this is also cross-reactive with NHPs. We use cynos in this study. They all have the same CpG domain, but they differ in their variable strand. And these 5 variable strands all came out of our platform through 2 main cell lines. One of them was for cancer cell line, for breast cancer, and the other one was for lung cancer. So we ended up with 5 different drug products. So these are 6 different single strand. This is quite heavy lifting for GLP tox. It was very expensive. It was a lot of work. And we synthesized all 6 strands in a bit lower purity than what we set for the clinical trial. And we pull them together, and we also established the analytical method to support this study. And we also characterize thoroughly the impurities of each one of them. So we can say that they are similar in terms of classes, N-1, N-2, POs, all kinds of protecting groups that are being deprotonized, and all kinds of things. We classified for each of them and then for all of them together to show both for our development process and both for the tox what are the levels of impurity and what are the levels of the classes of impurity, because eventually it's not a sequence manner thing, because we're going to have different sequences, but we did want to show that the classes are very similar across different sequences and we get similar response for them. The second challenge was which GLP tox bioanalytical method we're going to use. And we wanted to develop a method that could be used for the GLP tox and also for the clinical study later on. So again, what the problem is that we're going to have different sequences every time. So for the bioanalytical method, we actually took advantage of our constant part of our system, and more particularly is we use the CpG hybridization domain here. So we heated up our samples to temperature above our TM to denature this duplex, and we added great access of these 2 complementary strands for each side of the CpG sequence, each of them appended with a different dye. And then we monitored these 2 entities by fluorescent HPLC. And we were able to actually validate this hybridization fluorescent method for bioanalytical method, both for the GLP tox and also for the clinic in different matrices that we wanted to work with. In vitro safety. So again, this was a lot of discussion with the FDA, which kind of, let's say, even plans, because this is not like the preclinical data that we submitted. This is what we are planning to do for the patient data for the selection to prove that the sequences that are going to be introduced with are safe. So the first thing we have agreed on, and the FDA also agreed on, that we can't go through in vivo each time because it doesn't make sense in terms of timelines and even costs. So we can build some kind of in vitro prediction panel to try and assess the safety. And together with all the preclinical data and the GLP tox that was pulled together that we did, this will suffice the regulator in terms of safety. So we try to think of what can we do to say and to prove, or like to get like the best prove that we can, that these new sequences that we're going to manufacture every time that we haven't seen before and we know nothing about are going to be safe and to minimize their off-target effect. So the first thing is that our platform, because this is how the unique part, the variable part, this is its development, it actually has a built-in in it some kind of safety aspects because we do counter selection for the PBMC. So we actually choose sequences that we know does not kill the PBMCs, does not induce any toxic effect to this specific patient. It's not just to any PBMCs. So we know that in this aspect, in terms of, and we also did a lot of hemolysis for other samples that are just general samples for several candidates, just to show that the class itself and the protecting groups does not cause any agglutination or hemolysis. But we do know that for this specific patient, the sequences that we are going to choose are not toxic to its PBMCs. And then we also went through the literature and we found out that most, like other drugs, but also for oligonucleotides, are cleared through the liver, the kidneys, and are accumulated in the spleens. So these were the main organs, along with heart, that we tried to focus on and see that we will not have any toxicity towards these tissues, or not only cell binding. So we established these two panels. One of them was for toxicity, for liver, kidney, and spleens that we tried. And also we looked for sequences that are non-binding toward liver, heart, and kidney tissues. So these are not from the patient itself. This is just tissues that you can order and cell lines that you can work with. But this is something that we built that was acceptable for the FDA to show that in this part, when we synthesize individual aptamers that we need to choose from, and I'm reminding you that these are done in the final design with the same protecting groups in the duplex state with the T-cell engager part in it, then we screen to see which of them has the least toxicity towards these other tissues and does not bind any of them. Manufacturing control. So this was again a challenge because we needed to meet a set of criteria that will be flexible enough but still robust to be able to manufacture to some degree of purity that we need to decide on, and to have a set of specification that we need to meet for unknown sequences. And if you are CMC people, you know how challenging this task is going to be. So first we needed to actually decide what is going to be our set of specifications. Before that, what we did is we went shopping. We went through almost, I think, every manufacturer in Europe, basically, because this is closer to Israel, but also some in the US. And we tried to convince them to do this project to us. And it was a very hard task because we want them to actually dedicate a suite for us, because we can't even commit on a timeline. It depends on the recruitment rate of patients, and when do we get the biopsy, and when do we finalize the process. And usually if you want to manufacture, you need to book a suite like 8 months in advance, but we needed something much more flexible. And we also needed someone that was able to release a new entity for us every time, and no one was willing to take this commitment. So we ended up by building our own GMP manufacturing site. And the headline here says GMP-like manufacturing site, because eventually this was qualified for the phase 1 in Israel, which the requirements are not the same as what the FDA will require here in the US, or for anything that will approved here for clinical trials in the US. The requirements are a bit lighter. So it's still a GMP, but the requirements are different. So I added the GMP-like. So we had an independent facility that did everything from raw materials to drug product release. We had C-suites for the single strands manufacturing and release, and we had a B suite and an A environment for the drug product manufacturing and release, an internal QC, QA, to review this whole process. So because we are dealing with a duplex, we took our set of specification mainly based on the regulation and what is acceptable from white papers for siRNAs. But this left us with some tests that needs to be done to the two different single strands and some tests to be done to the final drug product. And this is quite heavy lifting. It's a lot of tests to do for each specific drug product that we're going to produce, because usually a clinical trial will have something like one batch to synthesize, and we are going to have to do between 15 and 20 batches for our only first in human clinical trial, which was a lot of things to develop and to do and to release every time. But in order to meet these specifications, we had to develop a process that will be flexible but yet robust enough. So for the constant strand, it was not a problem. It was very well characterized. We had the process, we know the impurities, we know it's the same every time. But for the variable strand, we were very worried because we set some kind of impurity criteria that we need to meet. And we ended up with some number and a very overkill process in order to meet these numbers. So our manufacturing process actually consists of a synthesis, two-column purification, IP and ion exchange, UFDF, and lyophilization that will actually make sure that we're going to end up with the adequate purity for an unknown sequence every time. And of course, the QC that needs to accompany each one of these steps in terms of identity, purity, concentrations. We then took these materials and we released them based on the monograph I just showed you. We took these materials to our GMPs, to our drug product suite, solvate them, hybridized them. We took them to fill and finish, which was also in-house, and released them. Most of the release tests were also done in-house. So in terms of the methods, again, we had to have methods that will be adequate for unknown sequences. They have the same length and the same modifications every time, which made things easier, but they have different sequence every time. So we actually ended up with being able to develop method both for QC and for production that were suitable for around 20% of the sequences that we tested. But the rest 10% wasn't working good with this method. And what was unique about this 10% is that most of them actually contained let's say unequivalent base composition. Instead of having about quarter of each base, more or less, we have a lot of A's or a lot of T's in the sequence, and they didn't work as well with this method. So we ended up with developing a second set of method for everything, IP, RP, and the QC, just for these sequences. And our process was that right after synthesis, we took a small amount of the crude material and we tested it just with this QC system, just to find out which one of these two sets we are going to use, both for the manufacturing, the downstream purification, and for the QC release. So we were able to actually meet this whole cycle within 12 weeks for phase 1. Okay, so I won't keep you in suspicion. I will show you some of the things that we came out of from our first in human phase 1 that was done in Israel. This was also presented, I think, in the last ASCO. And phase 1 was an open label dose escalation study. It evaluated safety and tolerability of our test item. We mainly looked for safety endpoints, but we also wanted to have preliminary evidence for clinical benefits and some PK data or biomarkers identification. And if we can also see something with the tumors, this will be very good. The delivery was local delivery, intratumoral, subcutaneous near the lymph node, or some what we called peritumoral, which is like subcutaneous in the tumor area, if the tumors weren't very accessible. The patient population was locally advanced metastatic, only solid tumors. We had a total of 10 injections. The first 4, the Those limiting toxicity period were 1 week apart, and then the other 6 were 2 weeks apart from one from the other. This was a dose escalation study. We ended up with enrolling 12 patients that have a variety of solid metastatic cancers. Most of them were heavily pre-treated, and we got them very spread and with very exhausted immune system. In terms of safety profile, we got up with a very good safety profile. There were no DLTs reported. Most adverse events were mild, and we had 7 serious adverse events, but only 1 of them was possibly related to our drug product. And the DSMB that we put together after every dose actually cleared all 3 doses for this clinical trial. In terms of clinical evidence, so for the overall radiology, we had 3 out of 11 evaluable patients achieve best overall response of stable diseases. One of them even you can see here the first one of them had more than 10 months of stable disease, which was the time that he was a part of the study, and then we stopped monitoring him. And if we look only at the injected lesion, we actually had 3 out of the total 13 injected lesion across all patients that had size reduction. This is our poster boy. It has a leiomyosarcoma on the skull, and it actually achieved a 40%, more than 40% reduction in its tumor size. And what was also interesting about this patient, and that it had 2 different lesions. You can see them here. We only injected to 1 of them, but we were also able to monitor and see size reduction of about the same for this uninjected lesion on its skull. For clinical proof, we had blood samples from all of our patients, which we were able to show T-cell engagement for almost all of them across all injections. And we had 3 brave patients that were also able to provide us with a biopsy after 5 weeks on the treatment. So all patients provided us with a baseline biopsy because this is what we took into our platform. But most of them didn't give us a biopsy during our treatment. Only 3 of them did that. But when we look on this biopsy and the tumor microenvironment, we were also able to see increased T-cell infiltration for almost all 3 patients and APC stimulations for these patients also. So I think that overall, the most important things from this talk is that it's highly recommended to reach out and communicate with the FDA as early as possible to pave the development pathway together, especially if you have something which is out of the box. And I think a lot of the new entities that are being developed today doesn't really fit into any classes that are, it's not like specific ASOs, it's not specific siRNA, it's not something that has this approved protecting groups, or even, I don't know, new backbones are introduced today. So once you're going to do something that is different from what already approved, it's best to reach out to the FDA as early as possible. We were actually invited to keep interacting with them through this pre-IND process and just keep submitting new pre-IND so they will be able to correspond and give us feedback. This was done during COVID, this development, so most of the feedback was actually done by correspondence. Use of multiple exemplary sequences. This is basically the most important thing for individualized therapy because you're going to end up with a new sequence every time. So the best way to prove everything you want to prove or to establish method is just to try as many exemplar as you think feasible to show that they are safe, they are similar to one another. This is from like platform design processes and principle that already have regulation for platform or some kind of suggested regulation for platform therapies. And invest in establish robust yet flexible manufacturing process to accompany analytical method. This is also something that a challenge that every individualized therapy developer is gonna encounter. This is it. Thank you for your time. I'm happy to take any questions. This is also my email if you have something later you would like to discuss.
Thank you, Zohar. That was fantastic. Are there... Seppa, we lost your video. Are you with us?
Yes. Let me quickly. So I'll kick off with the first question, maybe also a slightly difficult one. For this kind of platform approach, how do you go about specifying acceptance criteria specifically for both the enriching and as well as the manufacturing when it is deemed acceptable and when it's not good enough, especially if you're making a new one every single time?
Yeah, so I'll go back maybe to this. So again, this was a challenge. I think in terms of the specific set of criteria, this was actually not such a hard question because there is a lot of data out there and about what's the purity, and we know what's the purity for siRNA, which are about 20-25 bases long, and our system was much longer, but it was similar to what I think single guide RNA are today. So we were able to come up with a number somewhere around 80%, and then we improved our methods. So we even went lower to around 75%. That was acceptable by the FDA. So in terms of the numbers that we want to meet, it's okay. But we did say that we can't. We manufacture a number of entities, and we were able to show that we can get this purity, but we will include in what we call the FLP, the N-1, the PO. So there are certain impurities we know we cannot separate. And we can also not qualify by MS because it's going to be new every time. So we did get to some understanding that there are several impurities that are going to be under the main peak, which we also assume are going to be active, but they're going to be a part of it. We did set some limitations to this method. In terms of manufacturing, so this is why we ended up with 2-column purification. I can say now, after we did our first in human, and we did synthesize a lot of entities for this one, because we also had some patients out of the 12 that didn't make it to the 12 weeks endpoint. So we ended up synthesizing their sequences, but not injecting it to them. So we had more than 12 patients starting this process. And that we can now do it even in a single column and still meet the required purification, but we did ended up with doing a synthesis about twice the size that we're going to end up. So we can actually choose a very narrow set of fractions that will be much more pure than general manufacturing processes. And we also, for the phase 1, we did 2-column purification for each strand.
Can I ask a question about the regulatory discussions you had on risk benefit? I'm
Yes,
just wondering, you know, how did the regulators think about that in this context, and if there's any sort of lessons or takeaways that we can sort of channel towards individualized rare programs?
So I think that the most important lesson is to reach out early. I mean, early as when you're starting to think about your GLP tox design. This is because I think in terms of cost, this is going to be the after preclinical development. GLP tox is going to be the next thing, is going to cost so much. So this will be a good time to see that your plan is good and that whatever materials you're going to work with and the specification for these materials and the method to analyze these materials and also the bioanalytical method and what kind of pharmacodynamics and pharmacokinetics you're going to develop only for the GLP tox. This is even before the clinics are adequate in terms of the regulator. But I do find it both very positive and very helpful. I mean, they were very positive there. We actually submitted something which was very preliminary, and they gave us very positive feedback with a lot of suggestion of how to go forward in order to meet the required criteria in terms of regulatory. And they actually asked us to submit another pre-IND after we are, let's say, cook these ideas a bit more with us and have a more established methods and a more established idea of what we are going to do. And then we even had a third one. And we were even invited, you probably can find it online, we were invited to give a talk at the DIA FDA conference that takes place every 2 years about these strategies that we came up with together with the FDA and about this platform. So, yeah.
Thank you. And then when you initiated the program in Israel, did you find that all of the interactions with the FDA kind of led you to a point that held up there?
Yeah, but this was something we agreed upon upfront with them, because Israel is a very small place and they don't have the resources or even the expert to review this kind of very specific platform for aptamers and immunotherapy and personalized therapy. So we agreed that we will do all the process with the FDA, and then we just compiled everything and we submitted all this correspondence and all the FDA notes to the Israelis, and only when they saw that it's acceptable by the FDA, they approved it in Israel. But it was based on the FDA notes, which was something we knew upfront. This is how it's going to be done.
Understood. Thank you.
If there are no further questions, we will end the session here. Thank you everyone for attending. You are very welcome if there are any further questions or the discussion to interact on connections. Thank you again, Zohar, for presenting your work today. And I would just quickly want to give a reminder to fill in the survey, which will pop up at the end of the session.
Thank you very much.
Thanks, everyone.